All right, folks, here we are for our virtual lab number 4 on gastrointestinal physiology. This can be a little bit tricky just because we're using a lot of procedures and chemicals, but the overarching principles are pretty straightforward, so I'm going to try and make sure that you understand those overarching principles. Okay, so here we go. We're going to review some definitions. We're going to go over those overarching principles, and then we're going to actually get into the weeds of the lab with some test tubes and hot plates and enzymes and fun stuff like that. Okay, make sure that you review some definitions. Let's see, what color shall I use let's go with orange. I like orange. Make sure you review the definition for digestion as it relates to our digestive system. We may recall from lecture, digest means to break down. So we bring in big chunks of food, but we want to break them down into smaller pieces, and that is called digestion. We can perform digestion two ways. We can do mechanical digestion, which is what the oral cavity does, and the stomach, which is to say that we break down through mechanical means, like chewing, or like the stomach churning. We can also perform chemical breakdown of food, which is to break down food using chemicals called enzymes. Chemical breakdown, to chemically break down food through the use of enzymes. Then make sure you review the definition for absorption, which in the terms of the digestive system, that means we move broken down nutrients into the bloodstream. Movement of broken down nutrients into bloodstream. So this illustrates another point. We have to break down the food first, and then once our big chunks of food have been broken down into their smallest pieces, then we can absorb them into the bloodstream. That should be a review from lecture. Never hurts to hear it more often, though. And lastly, make sure that you review some things about enzymes. So we know that enzymes are chemicals. We know that enzymes are needed to perform chemical breakdown. But let's be more specific. The definition for an enzyme is a chemical that catalyzes or speeds up. Catalyze is just a fancy word for speeds up. So enzymes speed up chemical reactions, make them happen faster. Now, chemical reactions technically would still happen without enzymes, right? So technically, chemical reaction A or chemical reaction B in the body could still happen without the enzyme. But that's not realistic. We need to perform a lot of chemical reactions over and over again, so we need to perform them quickly. And the way that we can perform chemical reactions quickly is through the use of enzymes, because enzymes speed up chemical reactions. Then we have some characteristics of these enzymes. Enzymes can be reused, which is a great thing. So amylase can enter a chemical reaction, help speed it up, and then be released. And then we can reuse amylase again to speed up another chemical reaction. So it's very efficient. Typically, the name of enzymes ends in the suffix ase. So we're going to see in today's lab, we have amylase. We have lipase. What do you see that's the same for both of them? They end in the suffix ase. A-S-E. And lastly, you should be reviewing all of these, by the way. Lastly, enzymes are specific to each substrate. And what that means is amylase can only work on starch. So each enzyme, take for example amylase. Each enzyme can only speed up a chemical reaction involving a certain thing. They're specific, like a lock and a key. A key can only go into a certain lock. Amylase can only speed up chemical reactions involving starch. Lipase can only work on chemical reactions involving lipids, and so forth and so forth. But they are specific. All right. Good review. The more that we can hear these things, the better. Now, this is the slide that breaks down the whole lab. However, you're not going to be able to get away with just listening to this and then cutting out. You're going to have to listen to the whole video to be able to answer all the questions that you need to and to be able to prepare for your lab quiz. But this is still an excellent way to see an overview of what we're going to be doing virtually. So we are going to take a nutrient, be it starch, a lipid, or a protein. Let's start with starch, because that's what we're going to do in our experiment. We're going to start with starch. To that starch, we're going to add an enzyme. What is the enzyme specific to starch? Amylase. So we're going to take a test tube, fill it with starch, and we're going to add the enzyme. And then we're going to wait. And over time, what is that enzyme going to help to do? Chemically break down the starch. Okay, so we take the nutrient, we add the enzyme, and we wait. And eventually, we're going to see what that nutrient is broken down into. Starch is a complex carbohydrate. So, of course, amylase is going to help chemically break down the complex carbohydrate into a simple carbohydrate. Remember, we start off with big nutrients and we break them down into their smallest pieces. More specifically, this is going to be glucose, the monosaccharide. And we're going to repeat the same thing. Start with a nutrient. Let's talk about lipids. We're going to start with a nutrient. We're going to add an enzyme. In this case, we're going to add lipase. And then we're going to wait. And we're going to see what the lipids get broken down into. Lipase is going to help speed up the chemical digestion of lipid into its smallest pieces. We may recall from chapter 2 of this lecture, we break down lipids into fatty acids and glycerol. We're going to be focusing on the fatty acids. So we start with a big dietary fat or lipid, and we break it down using lipase into its smallest part, which is a fatty acid. Protein should be an easy one for you, because I know you know the structure of protein so well. We're going to take protein in a test tube. We're going to add the enzyme. In this case, the enzyme is protease. And then we wait, and we wait to see what protease is going to help speed up the chemical digestion of protein down into protein's smallest parts, which we know are amino acids. This, ladies and gentlemen, is the overall thing of what we're doing in this lab. Taking a nutrient, adding the enzyme, and waiting until that big nutrient that we started with is broken down into its smallest structural part. To help us identify when certain nutrients are present or not, we're going to be using indicator solutions. And we use these same indicator solutions in lab number 2. Remember, this is not the enzyme. It does not help chemically break down anything. What it does is it changes color. So we can add a certain indicator and it changes color of the solution. And then based on what color it turns, we can identify whether a certain nutrient is present or not. You do not have to know these indicators by name, but I'm going to write them down here. And when we go through the lab, I just want you to be able to identify that they're indicators, that the reason why we're even using them is because they help change the color of the solution so we can see whether or not a nutrient is present. For the lipids, we're going to be using something called litmus cream. And for protein, we're using a really long chemical name that I cannot pronounce, but its shorthand is BAPNA. Love the shorthands. Let's jump right into it, folks. So we're going to go in this order. We're going to start with starch, then we're going to do lipids, then we're going to do protein, and then we have another added bonus that deals with digestion and surface area. Here's where it gets a little tricky, but follow along with me. I'm going to try and break it down as best I can. And remember, you can always rewind the video. The wonders of technology. Okay, so we're going to start with starch. Okay? We're always going to do a control experiment first. We know from lab number 2 why we do a control first. We do a control to make sure the chemicals are working properly. And also, we do a control to see what we expect to see. In this case, we're going to expect to see certain color changes. So we do the control to make sure the color changes happen as they should. And then it lets us know what we're going to be looking for later. So what you're going to do is, in this lab, you would be adding starch solution. It's usually kind of a light blue color. You're going to be adding this starch solution to a test tube. So in the lab, if we were in the lab, you would make a mark at the 2 milliliter, excuse me. Or actually, I take that back. You would be using a pipette, a disposable pipetter, and you would take some of that starch solution and you would be able to suck up 2 milliliters worth of the starch solution. And we would add 2 mils of the starch solution into a test tube. Then you're going to add 2 drops of the iodine. So I put in 1 drop of iodine and 2 drops of iodine. And then I look to see the color change. Is there a thing on the next slide? No. Okay. When you do this, the color change that you're going to see, it's going to look like this. So once the iodine mixes with the starch, it's going to turn this color. Looking at the key below, because iodine is our indicator, based on this, is starch present? Absolutely. But it should be present. We just added starch. We added starch, and then we just added iodine to make sure that our indicator is working. When iodine is in a solution that contains starch, that solution will turn dark blue or black. So our control has worked. Now that we've done our control, we're ready for the actual experiment. What we'd be doing in the lab is we would be taking 5 mils of the starch solution and placing 5 mils of the starch solution into a test tube. And it tells me to label this test tube because we're going to be working with a lot of test tubes. So we better label it. So to our test tube labeled number 2A, we added 5 mils of our starch. This is what we said we were going to do at the beginning. We add the starch. And what are we going to add next? The enzyme. What is the enzyme for starch? Amylase. So now I'm going to add 2 mils of amylase. So we take the starch plus the enzyme. We would stir the test tube and we would record this initial color. Just so you know, the initial color of this test tube is going to be, it's still going to be kind of a light blue. The initial color is going to be a light blue. Okay, so we have the starch plus the enzyme. Now we're going to take this test tube and put it into a warm water bath. That warm water bath looks something like this. So we would actually take those test tubes and place them right into these little racks. And we can set the dial here, we have it set to 37 degrees Celsius. The reason why we incubate these in a warm water, by the way, this is about, 97 degrees Fahrenheit. The reason why we incubate these in a warmer environment is because this approximates the temperature of our body. What's our core body temperature? 97, 98.6? So we put these test tubes with starch and the amylase enzyme into a warm water bath that is the same or similar temperature to what our core temperature is in our body. And this is where the digestion is actually happening in our intestines, right? So what's the temperature in our small intestine? About 97, 98.6 degrees. So we want to mimic that in the lab. Another important point is that there is more enzyme activity at a higher temperature. So these enzymes will be a little bit more active at 97 degrees Fahrenheit than they would be at room temperature. So it's actually beneficial that our core temperature is a little bit warm because enzymes will work faster. Now, obviously, if our internal body temperature gets to be too hot, enzymes don't like when it's too hot. Okay, but at a moderately higher temperature, enzymes will work better. Okay, great. So, overarching principle. We started with starch, we added the amylase, put it in an incubating warm water bath, and we wait 30 minutes. Through the magic of technology, we're going to take that test tube 2A out of the warm water bath. And its color now is probably going to be maybe a little bit darker blue. But I don't want you to confuse this with a dark blue. Let's just keep it at that same lighter blue. In the lab, it'll actually be a little bit darker, but not much. So we're going to take that test tube out. And it's still going to be a light blue. All right. Now we're going to make a new test tube, 2B. And to that test tube 2B, it's not written here, but you're also going to take some of the solution from test tube 2A to test tube 2B. So into a new test tube labeled 2B, we're going to add 5 mils, and I apologize for the typo. So we take the solution from 2A, and we add 5 mils of it to 2B. But then to 2B, we're also going to add the iodine. So now I'm going to add 2 drops of iodine to the 2B. And I'm going to record the color. What is the color of test tube B going to be? This light blue. All right. Now I want to make a point here. In our control, we had a color of dark blue or black. Then we put that starch and we add amylase, and then we wait, and the resulting color is no longer dark black or blue. It's a lighter blue. So I want you to record this in your lab, and this will your lab assignment, and I want you to think about, is starch present? You can use the same color chart as before, and you can answer is starch present, and then you can answer why. Why either it is or it isn't. Okay. All right. So now we're going to continue on. So we've done the first two parts of our starch experiment, but we're not done, folks. We've got more to do. So now we're going to take another test tube and label it number 3. And to number 3, because we're going to be doing a control experiment, to number 3, we're going to be adding 2 mils of maltose. Maltose is a type of sugar. It's a simple sugar. The starch was a complex sugar. So our first control we did with starch was a control testing for complex sugar or starch. Now we're doing another control on a simple sugar. Okay? So I'm going to add 2 mils of my simple sugar control or maltose to a test tube. To test tube 3A, let me actually do the same colors before. To another test tube, 3A, I'm going to add the starch solution. So to test tube 3A, I'm adding starch, which is a complex sugar. So I know with all these names you might be getting confused, but I encourage you to listen to what I'm about to say. Let's simplify it. In test tube 3, we have a simple sugar, maltose. In test tube 3A, we have a complex sugar. We're going to add Benedict's, which is a reagent, to both. Benedict's is a reagent that tests for simple sugar or glucose, simple sugar. So we're going to add 1 ml of Benedict's to both test tubes. So we started with the test tube, one with simple sugar and one with complex sugar. Now we're adding an indicator solution that tests for simple sugar. Which test tube should test positive for simple sugar? This one. Benedict's, because it tests for simple sugar, it should test positive in our simple sugar control. But it should not test positive in our complex sugar control. Let's see what happens. Benedict's, as we saw in lab number 2, Benedict's requires a boiling water bath. So we're going to take both of these test tubes. We have test tube 3 and test tube 3A. We're going to place them both into a boiling water bath for 2 minutes. We're going to take them out, and then we're going to observe the color. I'm going to peek ahead to the next one. Okay. So we're going to have 2 different colored test tubes. Let me draw them out because you're going to need this for your lab assignment. Here we have test tube number 3. And here we have test tube number 3A after putting them in the boiling water bath. Remember, test tube 3 had our simple sugar. Test tube 3A had our complex sugar. So test tube 3 is the simple sugar. And test tube 3A was our complex sugar. What color will these turn after 2 minutes in that boiling water bath? Hate to say it, but I know because I'm a dork. The complex sugar is going to stay a light blue color, but the simple sugar is going to turn red. And the Benedict's is a test for simple sugar or glucose. So, does test tube 3 test positive for glucose? Very high levels. Does test tube 3A test positive for glucose? No. So our control worked. We expected the simple sugar to test positive for simple sugar. And we expected the complex sugar to not test positive for simple sugar. Perfect. Our control worked. Now we're going to do our actual experiment, part 4. Now, we're going to make a new test tube labeled test tube 4. To make our new test tube, we're going to be needing some of the solution from test tube 2A. If we go back, our test tube 2A was the starch plus amylase. Our test tube 2A is starch plus amylase, plus the enzyme that was put into a warm water bath. Okay? So we took that starch and amylase out after it was in the warm water bath. And just as a reminder, this test tube 2A tested negative for starch. It was no longer dark black or dark blue. We're going to transfer 4 mils from test tube A to a new test tube called test tube number 4. And then to our new test tube 4, we're going to add Benedict's, which is the reagent for simple sugar. We're going to add 4 mils of our Benedict's reagent. So to put this in simpler terms for you, what we have here is a solution of starch, which is a complex sugar, plus the enzyme amylase. We put it into a warm water bath for half an hour. And when we pulled that test tube of starch and amylase out from the warm water bath, it tested negative for starch. Now we're making a new test tube, and we're going to test this new test tube for simple sugar. Do you think it's going to test positive for simple sugar? Yes or no? Think about what starch gets broken down into. Now, of course, Benedict's requires the boiling water bath. So we put our new test tube number 4 into a boiling water bath for 2 minutes, then we take it out. And what is that solution going to look like? This is test tube number 4. It's going to come out of that boiling water bath as this color. You can look at the chart and you can determine whether or not starch plus amylase, after time in the warm water bath, after it tested positive for simple sugar or glucose. As always, if you have questions, email me, send me a message on Remind. I'm here to help you. Okay, now the lipids and the protein experiments are going to go much faster because they're a little bit simpler in nature. So we're going to do our test for lipids. Now we have a test tube labeled test tube number 5. The fun is still continuing, folks. We're going to add 3 mils of something called litmus cream. Litmus cream contains cream, which is a fat. Think about the creamer you put in your coffee. That's a fat. And it also contains a litmus powder that is an pH indicator. So cool. For our control portion, we're first going to add lemon juice. When you add lemon juice drop by drop, at a certain point, it usually takes maybe 7 or 8 drops. At a certain point, this solution is going to turn pink. And because this litmus cream contains a pH indicator, it just so happens when a solution turns pink, it is acidic. So here our control has worked. We added lemon juice, which is acidic. We added lemon juice to our litmus cream drop by drop until it turned pink. This is showing our control is working. When the litmus cream solution is acidic, it turns pink. Now we're going to add sodium hydroxide. Sodium hydroxide is NaOH. This is an alkaline solution. It's an alkaline product. Now we're going to add sodium hydroxide drop by drop. And you're going to add sodium hydroxide drop for drop until the solution usually turns this kind of blue. Until it turns blue. And because of the pH indicator in the litmus cream, whenever something is blue and it has litmus cream in it, it is alkaline or basic. It's a really nice, easy indicator, that litmus cream. If that solution is pink, it's acidic. If it's blue, it's alkaline or basic. Love it. What's our procedure now? Okay, so now we're going to have another test tube labeled number 6, and then one labeled numbered 6A, number 6A. We are going to pour litmus cream, 3 mils of litmus cream into both. And remember, litmus cream contains the cream, which is our fat, so it contains fat, and also our pH indicator. Now we're going to add lipase to test tube 6 only. So this is going to be our lipids plus the lipase enzyme. Whereas 6A, we're not going to add anything. 6A test tube is only the lipid. We're going to put both of these test tubes into a warm water bath, again at about 37 degrees Celsius. Why? Because this approximates our internal core body temperature. After 30 minutes, through the magic of technology, we're going to pull out test tube number 6 and pull out test tube number 6A. What are the colors we're going to see? 6A, let me just go back to make sure I know what color it was. It was that light blue. Okay. 6A, let's just say it turns this color. 6A. Whereas test tube number 6 is going to turn this. So you can identify which one is acidic and which one is alkaline. Oh, I knew I was making a mistake, and I apologize. This is going to stay blue. I'm confusing different ones, and I apologize for that. This is going to stay that blue color. Okay, they were both blue going in, right? They were both blue going in. When they come out, 6A is still going to be blue, but test tube 6 is going to be a more pink color. And we may remember, test tube 6A was our lipid only, but test tube 6 was our lipid plus the enzyme lipase. Now I'm going to refresh your memory here because I think it'll help you understand what we're doing. Back at the beginning of this lab experiment, when we were going through a review, we said that lipids use the enzyme lipase. And what do we break down lipids into? Fatty acids and glycerol. I want to point out fatty acids. So we'll know when lipids have been effectively broken down when we have the presence of fatty acids. This should help you in understanding our results here. Which one of these was the lipid effectively broken down into fatty acid? So cool, isn't it? I'm sorry you can't do this in real life. I do want to mention one thing. When we're in the lab, we sometimes have students smell these two test tubes. When something is acidic, it might smell a little sour. So that's another indicator we can use. In this case, test tube 6 sometimes will smell a little sour. Okay, number 3 already. Boom, we're on to protein. Here we go. Now we're going to be on test tube number 7 and test tube number 7A. Okay. To both test tubes, we're going to add our indicator. All right. You know, I forgot to add something in here, and I'm so sorry. But it's okay. Moving to this online thing, we come across these problems, and that's okay. We're going to add protein solution to both tubes. So just add this as another little bullet point here that I forgot to add, and I apologize. To both test tubes, actually, see, you know, this is a good example of it being okay to make mistakes as long as we correct them. We're going to add the protein to tube number 7. Again, I apologize for the mistakes. I'm so used to doing this in person. Okay, to test tube 7, we're going to add our protein solution. Something that we know has protein in it. To test tube 7A, we're going to add distilled water. That's what you see here. Add 1 ml of distilled water to test tube 7A. Okay? So we're kind of combining our protein and our control together. So to test tube 7, we have our protein solution. To test tube 7A, we have water. To both tubes, we're going to add the BAPNA, which is the indicator. So we're adding the indicator to both. This is not the enzyme, the BAPNA indicator. It only helps us know whether a nutrient is present or not because of a change in color. We're going to add protease only to tube number 7. So here we have our protease enzyme. Protease enzyme. We're adding the protease enzyme only to test tube number 7. So test tube number 7 has our protein solution plus protease. Okay, so the stage is set. Test tube 7, protease plus the protein. Test tube 7A, water, and that's it. So we can have an idea of which one we're going to be looking for a change. We can have an idea about which one we're going to be looking for amino acids in. Okay, we're going to put both of them into a warm water bath, of course, around 37 degrees Celsius because it approximates our core body temperature. And then we pull them out. Let's review. We had test tube 7 and 7A. 7A only had water in it. Test tube 7 had our protein and it had our protease enzyme. And because we added the BAPNA, which is the indicator, we know what we're looking for. If it's positive for protein, and what they really mean here is amino acids. Because, remember, from the very beginning, what is the overall point of this lab? We take a nutrient, in this case it's protein, we add the enzyme, which in this case is protease. Enzymes speed up the chemical digestion of the nutrient. And what is protein going to be chemically digested down into? What are the smallest structural pieces of proteins? amino acids. When we pull these out of the incubator, test tube 7 will be this color, and test tube 7A will still look more like this. So which test tube showed amino acids? You can tell, right? And we can tell, was protein effectively broken down into amino acids in test tube 7? I would say yes. So what we just did is we watched chemical digestion happen right before our very eyes. Simple. We take the protein and we add the enzyme and we wait. And if we're left with amino acids, chemical digestion happened. Boom! And it was assisted or sped up by the enzyme. Yes! Okay. I know you're anxious to get through this. You can't wait. So let's get to our last part of this Lab 4 on GI physiology. This one is about surface area, and we're going to use starch as our example. Surface area influences how much digestion can occur. So we're going to test this theory and test the theory to figure out, well, how does surface area affect digestion? By using 3 forms of potato. We're going to have a whole potato piece, we're going to have a mashed potato sample, and we're going to have potato flakes. So in the lab, when we would do this, we would actually take a real sample of each. So one test tube, we would label it whole or entire, because it's an entire potato piece. Then we would label a test tube mashed. And lastly, we would label a test tube flakes. Can't we agree that we have different surface areas here? A whole potato piece, a mashed or squished potato piece, and then really small potato flakes. So cool. When we do this in the lab, it's important that we make all 3 samples an equal weight, because we're going to be adding an enzyme to this potato sample, and we're going to be waiting to see how quickly that enzyme helps to break down the potato. In order to be able to compare the whole piece to the mashed piece to the flake pieces, they have to be a similar weight. So that way we're testing equal amounts of digestion. If I had different weights of these samples, the enzyme would be acting on it differently, because one piece might be big, one piece might be small. The same amount of enzyme on a bigger piece, it's not going to be able to break down as much. So we have to have the same amount of enzyme being added and the same amount of weight of each food. So in the lab, what we would do is, because when we started, we would prepare our potato samples with 1-inch piece of potato that we're going to keep whole, and another 1-inch piece of potato that we're going to mash. But before we mash it, we weigh them. So we would weigh each potato piece on a little weighing boat. I cannot draw. We would weigh each 1-inch piece, and we would take the weights, add them up, and divide by 2 to find the average. So we have 2 1-inch pieces that'll probably weigh about the same because they're both 1 inch. And now that we have the weight, or the average weight of these 1-inch pieces, we would add potato flakes until we get that same weight. So now we have 3 samples of equal weights. We would then add these prepared samples to our test tube. The whole piece we would add in as it is. The flakes we would add in as they are. For the mash, we have to take our 1-inch potato piece and mash it so it becomes mashed. Usually in the lab we use a mortar and pestle, which you might be familiar with from a kitchen, to mash a little bit and break down herbs. Now, before we start the actual experiment virtually, what is your hypothesis? Do you think the whole potato piece will be digested the most? Do you think the mashed potato pieces will be digested the most? Or do you think the potato flakes will be digested the most? Take a minute to think about it and jot down or keep in your memory what your hypothesis is. All right, let's get to the actual science. So we have our whole potato, our mashed potato, and our potato flakes, right? So we added those potato pieces. Well, actually, if I want to be accurate, this would look more like just a whole potato piece. Then we'd have a bunch of flakes. All right, now we're going to add the enzyme. Now, in this way, it's similar to our first three experiments. We started with a nutrient, in this case, starch. We're going to add an enzyme, which is amylase. We're going to add the same amount of amylase to each one. Then we're going to place all three test tubes into a, you guessed it, warm water bath at about 37 degrees Celsius, which approximates the body's internal core temperature. If you missed that question on our lab assignment or lab quiz, you shouldn't. After 30 minutes, we're going to bring them out. But we're going to be testing for the breakdown product of starch. We start with starch. We add the enzyme. And what does the complex carbohydrate get broken down into? Simple sugars. So that's why, after we pull these three potato pieces plus amylase out of the warm water bath, we're going to test them for simple sugar. You may recall our indicator solution for simple sugar is Benedict's, which requires the boiling beaker of water. So we're going to take these test tubes out of the warm water bath. We're going to add 2 mils of Benedict's to each test tube. Then we're going to place each test tube into a boiling water bath for 2 minutes, because Benedict's as a reagent only reacts to boiling water. Or not boiling water, but that amount of temperature, 100 degrees Celsius temperature. When we pull them out, we're going to have a look. So here we have our whole potato piece. Here we have our mashed potato piece. And here we have our flakes. Let me tell you what we're going to find. We're going to find this amount of glucose, or this amount of digestion in the whole potato piece. We tend to find this amount of digestion in the mashed potato piece. And then we're going to find this amount of digestion in the flakes. So using the key here, because we're testing for simple sugar or glucose. So actually, all three had some digestion. We started with starch, and it got broken down to, with the help of amylase, it got broken down into glucose. But there's different amounts. So here you're able to judge which type of potato piece had the most digestion. Okay, so what are some take-home points? Oh, yeah, my lovely students. Enzymes are needed to speed up chemical digestion. Chemical digestion would still technically happen without enzymes, but they would happen so slowly that we wouldn't be able to keep up with the chemical reactions that we need in order for our body to work. So we need them in order to carry out our cells' functions. And surface area does influence amount of digestion. When there is more surface area, there is more digestion. And let's talk about why. If we think about our whole potato piece, our mashed potato piece, and our potato flakes, I'm going to use the color green for the enzyme. The enzyme has to attach to the starch. So I know I'm simplifying it, but we're going to be able to get the idea. Based on this whole potato piece, based on the surface area that is showing, I can only fit 8 enzymes. 2, 4, 6, 8. When the potato is mashed, it's almost like the surface area is kind of spread out more because we've mashed it. So now I can fit 12 enzymes. Because there's more surface area of the starch, I can simply attach more enzymes to it. And if I can attach more enzymes to something, wouldn't there be more digestion? Absolutely. And let's look at the flakes. Let's say I can attach 4 enzymes around each flake. Now, obviously, I'm not going to be able to draw all this out. But if I can attach 4 enzymes over each flake, maybe in this little amount of potato flakes, I have 80 enzymes I can attach. So as we spread out the surface area, we can attach more enzymes, which means we can speed up the chemical reaction more and do more digestion more quickly. This is really cool stuff. How might this influence us as athletes? Well, that means that we need to think about what we're eating. If I want to fuel for activity, but I don't need it right away, maybe I'll have a Cliff Bar. Cliff Bar is a solid, isn't it? It's like a solid block. So if it's an hour before my Ironman starts, and actually an hour before the Ironman starts, maybe even a half an hour before the Ironman starts, I did eat a Cliff Bar, because I will use that energy, but I don't need it immediately. So I don't need to have the quickest digestion. But what if I'm in a race, or you're a football player, or you're a soccer player? Let's take soccer. It's overtime. You got 10 minutes left in the overtime period, and you want the quickest energy available. You probably wouldn't want the Cliff Bar. You'd probably want something like Gatorade. Gatorade is a fluid, so it's got a ton of surface area. So if I'm entering the last 10-minute overtime period and I need the quickest energy, I want to drink something or bring in something that has a greater surface area, because I'll be able to digest it more quickly. And if I can digest it more quickly, I can absorb it more quickly, and I can metabolize it more quickly. I'm hoping there's some light bulbs going off in your heads, ladies and gentlemen. Don't forget what we started with. You should be able to know what we started with, what enzyme we used, and what the end product of digestion was. You don't need to know the specific indicators other than knowing what an overall function of an indicator is. Woo! Great stuff, everyone. Thanks for working hard. Keep it up. I'll see you for the next lab.